So good morning. My name is Kelly Sharp. I'm actually at UCI and I'm in the dance department in physical medicine and rehabilitation at UCI. And today I will be discussing the visual and the vestibular system. Go ahead, James.
All right, so I'll go ahead and start sharing here. I think that should load now. So, good morning, everyone. So, my name is James Finley. I'm an associate professor in the Division of Biokinesiology and Physical Therapy at USC, also with appointments in biomedical engineering and our neuroscience graduate program. So, today I'll be kicking off our discussion slash tutorial about somatosensory contributions to the feedback control of movement. So to begin, I just wanted to provide a bit of a high-level overview of essentially my academic trajectory to give you some context about what I do and how this topic relates to the ongoing work in my lab. So I started my journey in the world of sensory motor control as a graduate student at Northwestern University. I did my undergraduate degree in mechanical engineering and have always been fascinated with understanding how complex systems function. And that really started from an early age with things like thinking about cars and how automotive systems work. And so when I joined the graduate program at Northwestern, I became really interested in this idea of feedback control and the world of feedback control during movement. And we'll discuss a lot of this today, but one of the most fundamental feedback circuits in the nervous system is the idea of a reflex or a stretch reflex. And so I spent most of my time in graduate school really examining how reflexes are modulated in humans to regulate joint level mechanics and how these reflexes change in different conditions when, for instance, we're dealing with unstable loads as a simple model of balance control. And so even when I was a student at Northwestern, my real interest with respect to research was really in the control of walking. And so I was able to transition into that world as a postdoc, where I did my postdoc at Johns Hopkins. A lot of that work focused on the idea of adaptation. So looking at how our walking patterns change when we modify features of the environment. So you can think about those modifications as a type of perturbation, and then essentially trying to understand the factors that drive those adaptive learning processes. So there is really when I started to move into the world of learning and adaptation in the context of walking. And then after that, I moved to USC, and essentially now where we sort of combine a lot of these ideas in the work that we do. So a common theme in our work these days is looking at how people deal with and respond to perturbations while walking. So now we use our treadmill to accelerate or slow down the treadmill belts, and we can look at how people respond to these perturbations, both when those perturbations are random or when they're predictable. And so we still really integrate these basic ideas that I started with feedback control, but now moving this all the way up to whole body dynamic movement. So since you all are here at IMSI, I thought I'd also just give a really quick sort of summary of a couple of really formative experiences for me that are somewhat similar to what you all are doing. So the first experience for me that was really formative was I was a grad student at Northwestern at the time. My advisor mentioned this summer school in computational neuroscience. I was very much a mechanics person, sort of moving into the world of neuroscience. And I said, sure, I'll go to this 3-week summer school. It turned out I would say I was very much sort of in the deep end, given that a lot of the topics were things I was not familiar with, but I think it was a really great experience. What I wanted to highlight is that I found some old pictures from these days, and you may notice at least a couple of people here, one of which is Simon Sponberg. So we both attended this summer school many years ago. He was, I think, the only other person who really thought about mechanics and neuroscience. And it's been really cool to now circle back. I haven't seen Simon in person in years, but to circle back and be involved in IMSI. Another experience was I was able to attend this workshop on time delay and balance control, which is very relevant for today's discussion. And this was, I think, maybe a 4 or 5 day workshop. But you may notice many of the faces in this picture, but two who I wanted to highlight. Of course, Lena was there, and also that was when I first met Madhu. So, the point is that as you all are involved in IMSI and meeting people across institutions, even as whether you're students or grad students, you may continue to really interact with these people throughout your careers. So just to put our today's discussion in context, I just sort of wanted to briefly summarize some of the things that we've been discussing over the past few weeks. So we started the first workshop really looking at this idea of isolated muscle experiments. And there was, one of the areas where we were introduced to the concepts like force length and force velocity properties of muscle, some of the limitations of classic experiments, and moving into this idea of looking at work loops to think about the dynamic function of muscle. From there, we transitioned to talk a bit more detail about computational modeling, and I thought it was a really great introduction to how we construct different types of models of muscle, or models in general, but models of muscle specifically, and then building this models at multiple scales. So that's, I think, a common theme across the world of modeling. At what level do you start, and how do you sort of build up or move down to understand more detailed mechanisms? From there, we talked in more detail about the idea of work loops, so using real data. So I thought that was a nice hands-on experience of how do you both compute work loops, so as Simon went through by hand, but also giving all of us the experience of computing those with real data. And then last week, our last workshop really finished with this idea of optimal control. And I think this is a really nice transition, because if you think about optimal control in the context of movement, particularly if we're sort of muscle-focused people, as I think most of us are, one way you can think about this is to try to answer the question of when and how much should a muscle be activated to achieve a given objective with some set of constraints in place. And so we've sort of worked our way to thinking about how muscle is activated, and I think that's a nice transition to talk about this idea of sensory motor control. So one of the things that you will see in the world of sensory motor control is this general idea that movement behavior emerges through what we might refer to as a hierarchical set of sensory motor loops. And the basic idea is pretty straightforward, right? We sense something about the world, we process that sensory information, and then we act. And the active behavior actually changes the sensory input that we have. We process that. And so we sort of have this endless loop. You'll see, of course, across the literature, people break this idea down into a more detail, where we try to now represent elements of the nervous system or specific sub-processes in this motor control strategy, whether that might be defining a target or desired behavior, executing that behavior, getting feedback, and computing errors. So one, I think, important point is that even when we see these types of engineering-like block diagrams, they are all a dramatic simplification of the reality. And it can either be overwhelming, or for me, I think really fascinating to just try to understand how this is all managed within the nervous system. So for today, I always like to start with some definitions of terms, because things get sort of confusing really quickly. These are working definitions, meaning I would not cite this slide per se, but these are ways that I think about a lot of these terms, just to sort of clarify ideas. All right, so we have mechanoreceptors. This is a pretty simple concept, but these are sensory receptors that are going to convert some sort of mechanical stimulus, usually a force or a distortion, so motion, into neural impulses. So that's what's going to, as we talk about all these different sensory systems, the sort of starting point are our set of mechanoreceptors located in our various tissues. We can think about then the signals that those mechanoreceptors transmit as our afferent signals, so going to the central nervous system. Later, I'll highlight what those pathways are, or at least a high-level view of those pathways. We can contrast that with efferent signals, where now we're really thinking about how information that's processed by the central nervous system and is sent back, through the periphery toward muscle. So those are, again, those control signals that we like to think about. I'll use this term processing. So processing is one of these, I think, fairly vague terms in the world of neuroscience. But now we can think about, we have sensory signals coming in, we have motor signals. These are just impulses from neurons. But as those neurons synapse on to one another, you can think about sort of math happening at those neurons, where you sum things or attenuate things or transform in different coordinate systems. So I'll just refer to all of that as processing. And so here's now where I think the main point of this slide is that when I think about sensation, right, I'm talking about all of those processes above, from mechanical transduction through processing, right? So sensation is to me is all of that. And that I think about that as distinct from perception, where perception is really more of this cognitive awareness or registration about your environment, right? So the bottom line here is that, sensation doesn't necessarily imply perception. So there's a lot of sensation that happens within the body that we're not consciously aware of. All right, so I'll be focusing on somatosensation today, which is this idea of body sense. And I won't talk about all the details on this slide, but what I want you to really take away here is that somatosensation has different elements of somatosensation. So we have proprioception as an example. That proprioception is mediated by different types of receptors that we'll talk about more in detail shortly. We also have this idea of what people might refer to as exteroception, which is the sensation of the external world. So when we sort of physically interact with our world, and we can think about essentially the sense of touch as being a dominant element that gives us exteroception, right? Heat, pain, things like that. Interoception is another part of somatosensation that we won't talk about today, but this is something, again, it's sensation that's not consciously perceived, but it's really important for giving the nervous system, the central nervous system, information about the state of our internal body. All right, so I think I just added this slide because there's a missing link in the story today. But this is just a table that I think is a nice way to sort of outline and organize the different types of receptors on the left-hand side that do the sensory transduction. So we can see here that we have a set of cutaneous receptors. And in the second column is noted the fibers that transmit that information, and we'll talk about why that's important shortly. And then in the final column on modality, you can think about the sort of more common terminology that we use to refer to the information that these receptors transmit. Now, the reason why this idea of fibers is important is because one of the, I think, really interesting things about physiology is that if you compare biological systems in terms of their ability to transmit, process information, they're actually quite slow relative to, human engineered systems. And so part of the sort of delay that is inherent in these systems has to do with how fast neural impulses travel down axons. So at the top here, where we have our cutaneous nerve, so this is, you can think about this as the nerves transmitting sensory information. On the x-axis, this is essentially a measure of conduction velocity, so how fast is that information transmitted, and you can see that all the way at the bottom. And then the different colors, the sort of dark blue, light blue, and orange correspond to different fibers. So the most abundant fiber type are these C fibers, but they are actually quite slow. So things like pain are transmitted really slowly. And I think we all have a real world experience with that, where if you touch something that's really hot, you touch it, and then there's a delay before you actually get the pain, right? And that's because the sense of touch is actually transmitted much more quickly to the central nervous system than that pain information. Then our fastest fibers are really the ones that are primarily located in muscle. We'll talk about some of those afferent fibers shortly, but we can also think about our alpha motor neurons. If we can maybe infer then that because the sort of nervous system has dedicated its fastest forms of transmission to muscle, that being able to quickly both sense and act is a crucial element of sensory motor or sensory processing. So when we think about somatosensation, we often start with the idea of touch. I won't talk about touch in detail today, but one thing to be aware of, if this is new to you, is that within our skin we have a set of different mechanoreceptors, and these receptors respond to things like pressure or deformation. And I think what's really interesting here is that, some receptors, they have different dynamic properties. And this is, I think, a common theme throughout sort of sensory processing. So we can think about receptors that are considered as rapidly adapting. So if we look at the figure on the right, anything that has RA in parentheses would be a rapidly adapting receptor. And so things that adapt rapidly, in other words, if I look at the bottom figure on the right, so if I have a stimulus that is a ramp and hold stimulus, these rapidly adapting receptors, they tend to signal information about changes. And so you could think about a change as something like a velocity, right? So when I sit on a chair and I feel, that transition from standing to sitting, right? I contact the chair. That's generally mediated by these rapidly adapting receptors. And then I stop, sensing that I'm in the chair. There are other receptors that are slowly adapting, which communicate more information about steady state pressure. And so they change and they continue to fire based on a sustained change in pressure or deformation. All right. So the next general idea here is we'll talk a lot about proprioception today, and this is really the perception of one's own body. And this largely communicates information about the configuration of the body. Proprioception is one of the sources of sensory information that provides kinesthesia or the sense of movement. And proprioception is largely mediated by, now we're sort of connecting muscle and sensation by sensors or receptors located in our muscles, tendons, joints, and skin. So now we're all the way back to muscle, and a lot of what we've really discussed so far, you can think about as having to do with the control of what we'll refer to as our extra muscle fibers. So when we talk about muscle, that's often what we're referring to. And so, as we all know, right, we think about our motor units as being those alpha motor neurons that lead to contraction of our extrafusal fibers. But within our skeletal muscles, we also have what are called intrafusal fibers. And these are essentially the sensory organ, or part of the sensory organs in muscle. So these intrafusal fibers, they have what we call their own innervation. So there's both a motor innervation. So here when we talk about the gamma motor neurons, so those intrafusal fibers can contract and relax. And then we have our afferent signal or our afferent connection that transmits information back to the nervous system. So if we look more closely at the idea of a muscle spindle, we have different types of different elements within a muscle spindle. We won't talk about all the terms here. The most important things to understand, and I have an asterisk here because we're going to come back to this idea later. But classically, we think about muscle spindles as having the largest afferent, so the 1A. They transmit information about muscle velocity, or primarily muscle velocity, as well as some information about muscle length. Whereas group 2 afferents primarily transmit information about muscle length. And so, again, for the engineers out there, if you're, let's say, an undergraduate student who's recently taken control systems engineering, one interesting thing that I found really fascinating about spindles is that, okay, now we have length and velocity feedback, which is something like, P and D when you think about PID control. In addition, the sensitivity to any sort of deformation, stretch, or velocity is modulated by our gamma motor neurons. So now we have a system where we can vary the sensitivity to different amounts of stretch. And so why might we want to do that? So let's look at our schematic of whole muscle again and think about what happens to the intrafusal fibers, so those in the sort of center of the muscle, when the muscle itself contracts. So when the muscle, shortens, the extrafusal fibers shortened, then if nothing else happens, those intrafusal fibers become slack, right? They haven't changed their activation. And so the problem there is that in theory, if those intrafusal fibers are slack, then they're not going to be very sensitive to changes in length or velocity. Okay? And so what we would like is some system that allows us to maintain to maintain the sensitivity of the spindle over different changes or different sets of muscle length. So how do we think about that? So let's now, again, I should have prefaced all of this. Most of the content that I'll be talking about, this is what I would call textbook level, sensory motor control. And what I, recommend for people who are really interested in this is to, look at some of the original work, but also to sort of question conventional wisdom. So some of what we'll talk about are basically oversimplifications of reality, and I'll point out one example of that later on with muscle spindles. Now, if we look at this illustration here, we have, just for visual purposes, the extrafusal fibers are separated here from the muscle spindle. And so now we can imagine that this fiber is communicating information about, let's say, muscle length. Okay, so the weight is added, stretches the muscle, the afferent fiber starts to send impulses. So now if we contract the muscle, so that's this idea of stimulating the extrafusal fibers, now we can see this, what I mentioned earlier, where the spindle is now slack. And so because I've contracted the muscle changes its length, and now the spindle actually stops firing here because it's slack, and then it starts firing again when the muscle contraction ends. So our gamma motor neurons, one thing that people think about gamma motor neurons is that they can be co-activated in some cases along with the alpha motor neuron, so that the sensitivity to muscle length is maintained as the muscle contracts. So now we see when the gamma motor neurons are active, we no longer have that drop in spindle firing. So now we can think about how this afferent information relates to muscle activation. So going from sensation to motor, so now we have a simple sensory motor loop. And the simplest loop here is what we call the spinal stretch reflex. So the stretch reflex is something that you all may have experienced if you've gone to your physician and they've sat you at the edge of the table and tapped your knee with a small hammer. That is a patellar tendon tap. That is something that triggers this stretch reflex. So one of the things that I think I thought was really interesting about this simple circuit is that we're basically triggering afferent signaling from the muscle. And so if I look at the figure on the right, so I have my muscle spindle here. Now you can imagine that if I stretch that muscle, that leads to activation of my 1A afferents, so going back to the central nervous system. And those 1A afferents directly synapse onto the alpha motor neuron that controls the respective muscle. And so you can think about this as something that we call negative feedback. So I stretch the muscle, that leads to contraction of the muscle. So you can think about that as a system that is at a very high level, trying to maintain the length of a muscle and keep that length even when there is a disturbance. So when I say this is a simple circuit, it's, I think, the simplest circuit because it's a single synapse at the sort of very simple, the simplest level. And now if we tie this back to the idea of our gamma motor neurons, so now you can imagine that if I modulate the sensitivity of those spindles, I can get a larger or smaller reflex response. And now I have this feedback controller that that's communicating information about length and velocity. So you can, again, classically, a lot of engineers have gravitated toward this type of system because you can in some cases, analyze it in ways that use some classic control theory. So in addition to muscle spindles, muscle, we can think about muscle, the muscle tendon unit also has what are called Golgi tendon organs. And these are mechanoreceptors located in the tendon. So you can see here, if I look at these 1B axons, they're integrated within the collagen within tendon. And what these afferents signal is what we call active force. So when the muscle is actively contracting, if we look at the force on the x-axis here, those afferents fire at a rate that's proportional to the force that the muscle is generating. Now, this force information is involved in another, I think, really interesting simple feedback circuit where the polarity of this feedback actually changes based on the task that you're performing. So now this diagram is a little bit more complex than what we saw for the stretch reflex. But the idea here is, again, I have my 1B afferents. Let's go here. And now if I contract the muscle, that afferent information comes back to the spinal cord. But the key difference from our typical stretch reflex is that now those afferents are they're going to synapse onto an inhibitory interneuron. So it's now not just a single synapse. So now you can think about this as a system that tries to prevent your muscles from, quote unquote, over activating. So if I have a really large force, then this system tries to bring that force down. So you can think about it as potentially something that might be protective. So if we look at what happens when those afferents are directly stimulated, so the sort of vertical line here represents stimulation of those tendon organ afferents. And then the data here are ankle extensor motor neuron firing. So typically, if I stimulate that afferent, that would lead to a reduction in activity from that muscle. And this is, again, a form of sort of negative feedback. However, when walking, if you do the same thing, now there's actually a change in the behavior of that circuit. And what happens is that this sort of what we might call gating, where now that afferent signal leads to an increased activity of that muscle. So again, the same stimulation here leads to an increase in the activity of the extensor, and that's what we would call positive feedback. So generally in engineered systems, we often stay away from positive feedback because it can make systems go unstable. But it turns out that this reflex is thought to be really important for maintaining anti-gravity support. So in other words, if my ankle extensors are active, that means I likely during gait I have weight on my leg. So I actually want to make sure that those muscles don't give out or go quiet. So that's one reason why people think this is an important source of feedback. All right. So when we think about experimental methods to study these circuits, essentially what we're talking about are perturbations. And if you look across the literature, the sensory motor proprioceptive circuits are studied using various types of perturbations, where we might think about things happening at the muscle level with the tendon tapper. A lot of my PhD work was looking at the joint level, and some other people in IMSI do this as well, where we might use a motor to rapidly stretch or flex or extend a joint that stretches those muscles, and we can measure reflexes through EMG. Classic studies by like Nashner, Horak, McPherson, and so on have used whole body perturbations, where now you can take a platform and either translate or rotate the platform to examine postural control. And so I might call this quote unquote static whole body responses. And then finally, our work and many others now are looking at perturbations of dynamic behaviors like gait, where we might accelerate a treadmill or pull on someone while they're walking. But still, this is just an evolution of the idea of using a perturbation to study sensory motor feedback control. So in the interest of time, I'll sort of skip this slide. This is just if you're interested, a way that you can think about sources within the nervous system where different types of processing can happen. Anywhere there's a synapse, now we can sort of have some processing. Now, because this afferent information, travels from the periphery through the cortex, you can think about this idea that as you go higher within the nervous system, now you can integrate that sensory input with other sources. And so, when I was a graduate student, one of the things that I was really interested in is this idea of long latency stretch reflexes. And one of the things that we know from really nice neurophysiology is that when I stretch a muscle or a joint in vivo, one of the things that you can measure is the activation of motor neurons in the cortex in response to that stretch. And then that activation actually can lead to a descending, efferent muscle contraction. And so even though this loop goes through the cortex, it can often be faster than what we consider to be a volitional response. And so I spent a long time in my PhD trying to, read a bunch of papers about these responses and how they could be modulated. But I spent, I think, about a year and a half trying to reproduce one of these responses in humans, and it turned out to be much harder than I thought. But it actually led to something that I think is really, really fascinating. All right, so on the right, we have, this is an actual reflex response from a human, so one of my participants from when I was in graduate school. So if I rapidly flex your ankle while you're maintaining some low level of force, we can get these reflex responses where the shortest response here we think is primarily spinal. But then there's these longer latency responses that might probe higher levels of the nervous system. Now, one of the things that I struggled with was that for a very long time, I was not seeing these so-called medium and long latency responses. And after looking across the literature at the actual perturbations that people were applying, one thing I noticed was that some of the perturbations were what we call very sharp, so have really large acceleration. That's what we had in the lab. Whereas other perturbations were really what we call torque controlled, so they were fairly smooth when it came to the acceleration profiles. So one of the studies of my dissertation was to examine whether the sort of dynamics of those perturbations affected whether or not these longer latency responses were present. So here we could vary the acceleration of those perturbations and ask how that varied these short and medium latency responses. And so the basic take-home point here was what I found was that the perturbations that we used with really high acceleration, so what you see on the top level here, those led to a really large short latency response, but you didn't really see much later on. However, if we started to smooth the perturbation, so now this is going from top to bottom here, we started to see larger responses in this MLR or medium latency window. And so the short latency response just became larger and larger as the acceleration increased. But the medium latency response really peaked at this sort of moderate level of acceleration. And so when I wrote this paper, I didn't really understand why, or if there was really evidence to support this idea that reflexes were maybe sensitive to acceleration. So we just were sort of guessing about potential physiological mechanisms underlying that observation. But I don't know if Lena is here today, but I remember talking to her several years back when I believe she was on sabbatical, actually recording information from muscle spindles. And some of that work turned into some, I think, really, really great physiology that started to help us rethink how muscle spindles communicate information, or what information those spindles communicate back to the nervous system. So these are a couple of papers related to this idea that things like force or derivatives of force might be encoded by muscle spindles, and that sort of the story that we've been telling ourselves is maybe not so simple. All right, so we have about 5 minutes left here, and I think we are, near the end. I'll skip over my own work. So one thing I wanted to make sure we we're able to talk about here is that I mentioned going from the muscle, we talked about the joint level responses, but a lot of information that we know about sensory motor feedback control has to do with perturbations at the whole body level. And this is where now we're actually in the world of, you might call it sort of comparative neuromechanics, where classically these studies have been done in cats, where cats are placed on a platform, that platform translates or rotates, and people have examined many aspects of how those reflex responses change based on the task and the direction of the perturbations. So one of the things that happens once you start moving to these whole body responses, that now you have muscle activations that are distributed across multiple joints. And so now we need to think about coordination across muscles and joints. And this coordination now happens at a much higher level. There's some that's mediated by spinal circuits, but we think now a lot about the role of the brainstem in coordinating these postural responses. And so as I mentioned, if we look, and this is again, this figure on the top right is, now we have this outline of a cat where we're looking at the response of the gluteus medius, so one of the hip extensors, where the now perturbations can occur in different directions. And so we can see that for the gluteus medius, right, the perturbations that are in this sort of anterior toward right direction tend to elicit the largest responses from that muscle. And this is this idea of directional tuning of reflexes. And so the same thing is true for many of the muscles throughout the body. So not only can you look at this tuning, but you could also look at how muscles are coordinated. So in other words, which sets of muscles tend to be activated together. So there's a whole world of research looking at those patterns of coordination. And then the last thing I'll show here is that when we think about these postural responses or even sometimes very low level reflexes, they can be tuned to both the sort of task constraints. So here we're showing what happens on the top when someone is standing on a narrow beam, and that beam is translated backward, where you see they mostly rely on the knee extensors and hip flexors to maintain balance. In contrast, when standing on on a flat surface, so on the bottom here, when that platform is translated, people rely more on the ankles and the posterior chain. So people refer to this often as the sort of hip versus ankle strategy. Now, if you take one and you have them practice on a beam for some time, they learn to use this hip strategy, and you then move them to a flat surface. It actually takes some time for them to switch from the hip to the ankle strategy. And so we can think about this as a form of what we might call sensory motor adaptation, where you by default are attempting to use a particular strategy. That strategy is no longer, let's call it, optimal in a loose sense for a given context. And so through trial by trial, maybe trial and error, you learn to adapt a strategy that's more appropriate for that context. All right, so to wrap up here, I know I've talked to some people who are interested in kind of clinical careers. Some of the reasons why we're interested in understanding these even sometimes simple circuits is that it's important for many different clinical populations. So for instance, people with diabetes often have peripheral neuropathy, where now the large diameter sensory fibers, again, those that innervate muscle, tend to degenerate. And so that can lead to postural disturbances. A similar thing actually happens in chemotherapy. So chemotherapy can lead to neuropathies as well. Diseases like multiple sclerosis lead to what we call demyelination, which initially affects the transmission speed of information, but can lead to things like debilitating pain and problems with coordination. And then finally, in people who have stroke or spinal cord injury, they may have things like hyperreflexia that can affect both posture, and there's some debate about the role of this hyperreflexia in movement that we can discuss maybe later on. All right, So I will wrap up with some what I call partially open questions. Really, things as part of IMSI, things that we might be really interested in understanding are, how are these reflexes modulated across limbs and in different tasks? What's the role of learning in the control of these reflexes or higher level responses? I think several of us who are part of IMSI have been really interested in questions about how reflexes affect both muscle, joint, and whole limb mechanics. So there's a large body of work that's really interesting in that space. The last two questions are, I think, some of the hardest questions, right? So how do we even think about what the nervous system is controlling? If you want to go down like a 40-year rabbit hole, there's an idea, it's called the equilibrium point hypothesis, which really, I think the idea is really interesting, right? The idea is that maybe the nervous system is just controlling the set point of muscle, so like the desired length. And once you control the desired length, then forces emerge from any deviations away from that length through these reflex pathways. So it's a very different idea than I think how we typically conceptualize motor commands. And then lastly, one of the big challenges in these closed loop systems, I think maybe Greg alluded to a couple of weeks back, is how do we infer cause and effect, right? So what does it mean for something to be volitional versus reflexive? And then how do we study these sensory motor circuits in real behaviors where there's sort of ongoing cyclic activity? So with that, I will wrap up. I think we're about halfway in, and maybe we can take, let's say, one question or two questions before transitioning to Kelly here. See, there's a chat. I was not monitoring the chat. Oh,
I think that was just some technical difficulties, troubleshooting. Any questions here? So James, do you have a take on how we should conceptualize control?
oof. That's a huge question.
Yeah.
Yeah. So this is, I think, an area where, so now we're kind of talking about conceptual models, right? And I think this is an area where we can be actually wrong, but have a reasonable conceptual model of how things work that allows us to explain behavior. And I'll give an example. I think optimal control is one of those ideas, right? Where, optimization is actually, in any kind of complex system, is extremely hard. And actually true optimization is basically impossible, right? There are too many degrees of freedom to really search and find the single best solution. But if we think about this idea that biological systems are attempting in general to find what I think Jerry Loeb refers to as good enough solutions, then that leads us to something like optimization. Right? It leads us to gradient descent, even if it doesn't lead to a perfectly optimal solution. And I think that does explain, we can use that idea to explain many things about behavior. But I do think it's a topic that's worth discussing, in the IMSI is, in the world of neuroscience, I mean, there are 50, 75 years of questions about, do neurons encode position or force or limb configuration. So these are really long-standing, I think, difficult questions to answer. All right. So I think with that, I'll hand it over to Kelly.
All right, thank you so much, James. We're now going to switch gears. Let me see if I can get everything set up. Can you hear me okay? Great. There is a fire about 2 miles from my place right now, so there's helicopters all around. So if you hear any noise, I apologize. Advanced, here we go. Give me 1 second. See if I can do this right. I learned this last night on YouTube. Can you see my slide? Okay, excellent. So I just learned how to do the notes and the slides at the same time. How many years have we been in Zoom? All right, so I'll go ahead and get started because I think I have about 41 minutes. So First of all, my name is Kelli Sharp. Introduced myself earlier. I'm associate professor. I'm in the dance department. I'm also the chair of the dance department. That's another whole conversation. And I'm the vice chair for research in physical medicine and rehab. I'll just go briefly. My career trajectory is very different, and I like to share it in the sense of making sure that people understand that there isn't one pathway. There's lots of different pathways to get to different spots, and it's a journey, right? And to embrace a journey. So basically, I grew up in Virginia. I watched animals and movement. I grew up on a farm, so movement has been part of my life. And I was headed and slated to go to medical school and totally disappointed my whole family and didn't talk to me for months when I didn't go to medical school and decided I want to take a career path in research. Came to UCI, studied spinal cord injury and looked at in basic animal models and really focused on axonal regeneration in the beginning and SCI models, and then worked my way into more of mechanics of how forelimb function occurs and hind limb function and what are assisted devices that we could create in the rodent models. And I'm very blessed to have worked with Dave Rinkesmeyer my whole career. And then I went and decided I wanted to work with some people. So I got a DPT at Chapman, and then I did a postdoc and did neuroimaging. So it just sets up this little story that we all have different pathways. And then I got really lucky, got a complimentary wellness grant, like right in while I was doing my postdoc. And that kind of set me to the future. And everyone asked, like, how did you get into the dance department? What did that look like? So interesting enough, all my work is based on movement strategies and learning with individuals with different neurological disorders. And someone from the dance department saw me give a talk in the stem cell building and thought the individuals in the wheelchair were dancing, even though they were working with a robotic device. And so I got in that way. And so looking at movement strategies. And so that's what I focus on right now, is using dance as a rehab tool across different populations with neurological disorders. And also the dance focus is really figuring out how do dancers learn and what's the best way to deal with their performance anxiety in these different learning environments. And since I got tenure, a little bit more freedom in life, I'm super excited that I work in adaptive sports with looking at psychosocial wellness. So as just want to acknowledge the land that we're on. A lot of us are at UCI, just really making sure that we honor this space. And I just realized that was an old slide from Wake Forest. Did not update that, so I apologize for that. But we're just going to go through two systems today in the basics, understanding the vestibular system, the interactions, the visual system, and how do we study these systems. So there's three main components that we've already talked a little bit. James went through the somatosensory system, and I'm going to go through the vestibular system and the visual system on a very basic level. But clinically and experimental design, it's really important to understand these basics. And especially if you're looking at the interactions between how we move, these sensory systems are really integrated into these design. So the vestibular system, we'll start here, is really just thinking about there's three main parts. Can you see my pointer? So you have the external ear, the middle ear, and the inner ear. We're going to focus more on the inner ear, but it's really important to think about what are the functions of the vestibular system, right? So gaze stabilization, ensuring that the field of vision stays focused when you have a subject of interest. And this is interesting when you're designing different experiments with people that have different peripheral deficits or changes, depending on what the disorder they are living with. Also understanding that body position and locomotion and our balance is really integrated into the inner ear, orientation of the body with respects to gravity, and our reassessment and readjusting of our autonomic functions occur with this body reorientation that occurs from this sensory information that's going into the inner ear. So in simple terms, the vestibular system is really driving responsibility for motion, head position, and our understanding of spatial orientation. For the sakes of this presentation, I'm going to break down the vestibular system into two parts. One is the peripheral vestibular system, meaning outside the brain, and then the other is going to be the central vestibular system. So here you can see in these three images, this is the most outer aspect. And then we have this vestibular apparatus here in the middle image. And then we have a image of what's happening actually in the utricles and the saccule, which we'll go into here in a second. So the vestibular apparatus is, you can see here in the top image, it's made of 5 organs, right? So you have 3 similar circular canals. And so here's one, here's another, and here's the third one. And then you have 2 otolith organs, which are the utricle, which is here, and then you have the saccule here. So in the semicircular canals, you have these things called ampules, and inside the ampules, you have these cristae. And we'll go and break down this here in a second. So in the utricles, they're really responsible for detecting horizontal movement, right? So what is happening in our head horizontally? And in the saccules, they're responsible for this vertical movement. And it's really important, as James was talking about, there are different types of receptors, right? So in the semicircular canals that we're talking about here, those receptors are called the cristae receptors. And then you have the macula receptors that are going to be in the saccule and the utricle. So it's important to think about what are the differences in the roles within those and understanding head motion. So let's break down what's in the cristae. And these, remember, the cristae are in the semicircular canals that are responsible for head rotation. So you can see here that here is these hair bundles, right? Here are the hair cells, here are the hair bundles, and this is what's called the ampule. And that's at the distal end of the semicircle. And the hair cells are really responsible for when there's actually a fluid in here. And when the fluid moves, it sent the hair cells move. And that is what causes the head, like the cupula move. They get disoriented. And then the fluid moves with head rotation. And so that's what's giving this input and sensory information that's going to be sent out of the semicircular canals. And then it goes down. And we'll go through the pathway here shortly. And it's really important to think about what is actually the physiology. So when the cupula moves, right, and that's this top part of this hair cell bundle, the entire population of hair cells, they become depolarized, and the activity innervating of these axon increases. And when the cupula moves in the opposite direction, the population becomes hyperpolarized and the neural activity decreases. Right. So you can see here, and this is just the bottom figure here is just an orientation of what is happening with the orientation of these cupullae within the inner ear. So the next thing I want to talk about is the macula. So the macula are the receptors that are in the saccula and the utricles. And they have a very similar makeup, but in the sense that the macula of the utricle lies in the floor of the horizontal plane. So you can see here, this is the horizontal plane, and then this is actually in the saccule, this is going to be in the vertical plane. These receptors, they sense the position of the head in response to gravity in linear acceleration. So that's important to think about when you're designing like even a perturbation study, right? Which are you looking for, vertical or horizontal perturbation? So the next thing is really thinking about, you can see in this picture here, we have the 3 semicircles, we have the cristae that are responsible. They have these like hair senses. And then within the utricle and the saccule, you have these afferent, and here's the macula here. And you have this blanket of calcium carbonate crystals that is really important to think about, because with these crystals, the crystals are calcium carbonate, they are called ostotonia, and they lead to movement of these hair cells. So when they become activated, when your head moves, the gelatinous mass moves across this calcium carbonate, and which stimulates the hair cells. So the other thing to really think about is after your head moves and the fluid goes through across and is activating the calcium carbonate crystals, then the receptors send impulses to the brain about movement and the specific canal that it is stimulated from. The other thing that's important is that balance information from these sensory organs, eyes, muscles, and joints are sending information to the brainstem, and we'll go through that. and the information is sorted out and integrated and then it's contributed to what's going to happen in the cerebellum. So in the central vestibular system, what we're going to focus on is let's talk about the different components. The different components are you have the cerebellum, which is here, you have the vestibular nuclei, which are in this area, and the pathways to the thalamus and the cortex, which provide sensation and perception of head movement. And we're not going to go too much, but it's important to think about the vestibular spinal pathways, which are motor commands to the muscles of the neck, the upper torso, and the upper limbs to maintain our balance and posture. And the other thing is that you also have the nuclei of the cranial nerves of 3, 4, and 6 that all control our eye movement. So the vestibular ocular reflex, it functions to maintain a steady image of the retina when the head is moving and produces these compensatory eye movements when you're moving your head in the opposite direction. So this is the central vestibular system. And let's break it down a little bit more. I would be amiss not to talk a little bit more about the cerebellum. So the cerebellum, we're just going to briefly discuss this, is the functional regions that are related to the vestibular system are the cerebellum, which is responsible for planning movements and motor learning. And then you have the spinocerebellum, which is body function and locomotion. And where the adaptation talks about, which it relates to what James is talking about, is the vestibular cerebellum, which is responsible for adaptation, compensation from vestibular deficits. Really important in this understanding of motor learning, and then also regulation of balance and eye movements. So moving forward, I think this is one of my favorite slides, and I actually love the 2-minute neuroscience blurbs. If you see the videos on YouTube, I recommend everyone to check them out for a quick review. So you can see here that we have the ampullas that are in the sacula, and in the utricle, we have the macula, and just really thinking about what are the general movements that the vestibular pathways are responsible for. And we're not going to go into it too much, but just understanding this is a very complex system. And when we're designing experiments to test some of these things, we need to think about what pathways are we actually affecting and what are the changes that we want to understand and see, in what order of processing are we trying to test. So the next thing is just really briefly going to talk about some really important reflexes that are really key components in the vestibular system. And we have the vestibular ocular reflex, which ensures the gaze stabilization. And that is really important for recognition and actually going to a point. So if you have someone grabbing a cup, you'll see if they have deficits in their vestibular ocular reflex, they actually won't be able to grab the cup. And vestibular spinal reflexes, which is really ensuring maintenance of an upright position. And the vestibular colic reflex ensures head stabilization in space. And there's really some beautiful work in babies that has looking at this colic reflex and changes over time and adaptation that takes place, orientation in higher species, and navigation of perception of self and position, and really understanding, like, the other thing that's important is thinking about our vestibular system is influenced by our circadian rhythms as well. So in summary, it's really important to think about the vestibular system in relationship to how it integrates with our postural control and our also somatosensory. But the other thing that we really need to think about from, going more from a peripheral system is really understanding the role of our visual system. And I'm just going to go through the basics of our visual system. So you can see here in this slide, here's a picture of an apple, and then we have an eye with an optic nerve, and then we have the lateral geniculate nucleus that then goes into the visual cortex. So let's break this down a little bit more. And it's really important to think about that it's a 4-step. The visual system is kind of a 4-step process. You have image formation from the cornea and the lens. You have exposure control, which is done by the iris and the pupil. And then we have photoreceptor sensitivity, which we'll talk about from the rods and the cones. And within the detection, which also occurs within the retina, which is considered like the sensory mecca of this processing. And then we go to the processing that occurs in the brain. So we'll just go briefly into the human eye. I think it's really important that we understand the mechanics of the eye, especially when we're talking about working with different disorders or designing experiments that have a visual component or proprioception input. So the eye consists of a pinhole, or what all of us know as our pupil, and which emits lights. And then the lens, right, is actually what focuses on light. And then we have photoreceptive elements in the retina that transduce the light, right? The retina is located in the posterior portion of the eye, and it's detecting the photons that enter the eye and then turning them into electrical impulses that exit out the brain through the optic nerve. So behind the cornea, it's important to think about we have what's called the iris, and the iris is the colored portion of the eye. And in the center of that, we have our pupil. And the reason why I bring this up is that contraction or relaxation of these muscles in the iris cause the pupil to constrict or dilate in response to various lights. There are different disorders that affects this actual contraction. And if you're working with a clinical population that has these contraction issues, their peripheral vision is affected, and you have to reassess and adjust what activities that one may be doing with them for that. The space from the back of the cornea and the front of the iris is this anterior chamber, and it's filled with this like aqueous humor. And it's important to think about that a lot of times as we age and with different disorders, we have a lack of this aqueous humor, or it becomes viscous. And I think it's important to really think about what is we need to know the basics of the eye to understand how to move forward with certain things that we're studying. So let's go in and think about like the visual pathway, and then we'll break down more of what the retina does. So you have visual receptors in the retina in this basic pathway. The first order neurons are bipolar cells in the inner nuclear layer. And then there is a second order neurons that are ganglionic cells in the ganglion layer. Then it goes to the optic nerve. Then it goes to the optic chiasm, optic tract, and then the third order neurons in the laterogenic nucleus body, and then it goes for optic radiation, and then the visual cortex. And we'll break this down a little bit more and explain why this is important. So here is another example, an image of here's what's going on in the retina, and then just breaking down what we just talked about. You have the ganglionic cells, the bipolar cells, and then we have our rods and cones. So let's really just break down the retina a little bit more. So you can see here's the fova, here's the optic nerve, we have the retina, and here are the blood vessels that feed the eye. Light passes through the pupil and then is focused on the back of the lens onto the retina. And the retina consists of three main cell layers. And this is important that we just went through. You have the ganglion cell layer, the bipolar layer, and then you have the photoreceptor layer. And this is what actually transduces light. But there are quite a few different disorders, and actually a lot of stem cell trials that are being implemented in aiding in this ganglion layer and then also in the photoreceptor layer. So it's important in the ganglion cell layer is the outermost layer, and the photoreceptor layer is the innermost layer. In order to reach this photoreceptor layer, light passes through the outer two layers into the retina. So let's look again. So here we have the optic nerve. We have the optic nerve, remember, is coming from the retina. We have the ganglion cells, the bipolar cells, and then we have our rods and cones, and then we have the pigment epithelium. So let's break this down. There are two types of basic photoreceptor cells. We have rods and cones. Rods are in the peripheral part of the retina. They're responsible for night vision, right? So some of us who are aging, I'm not going to say my age, but definitely feel that this is changing over time, and my peripheral vision is changing. Rod vision is black and white and low in resolution. And then it's basically a process called dark adaptations, where the rods can progressively increase the sensitivity to light and dark conditions. Dark adaptation is relatively slow process, and it can take several minutes. And as one ages, it actually takes longer. Whereas cones are in the center part of the retina, and they're responsible for colored vision. So for example, this is where they detect different colors of red, green, and blue. And the cones do not work well in poor lighting, which is why we progressively lose color vision as it gets darker. So it's important to see here, these are cones and these are rods, beautiful image. And thinking about there are like 120 million rods to 6 million cones. So you think about the actual adaptation that can occur between black and white and color. And then the other thing to think about is this general like transducts, like what's happening. So here is just a quick overview. Again, we have the photoreceptors, right? We just learned that we have rods and cones. We have the bipolar layer and the ganglia layer. So let's break that down a little bit more. So visual transduction is important for understanding like these photopigments are located in the membrane of the outer section of the rods and the cones. And each pigment consists of a protein, opsin, and retinol, which is a lipid. And in the dark, this membrane sodium channels are open and glutamate is released, which depolarizes the membrane. And then the light splits the opsin in the retinal apart, and you get this activates transducin, a G protein, and then activates this phosphodiesterase, which reduces cyclic GAMP and closes these sodium channels. And this is important because there are actually different disorders that affect this closing of the sodium channels that can, if you're working with different patient populations, can affect how you design your experiment, especially if you're doing something. I learned this the hard way. I was doing an experiment in an MRI machine and I had secondary comorbidities is that 6 of my patients had issues with their cyclic AMP production, like reduction in their eyes. And it was like they couldn't see the image that was in the MRI machine. So it's really important to think about these things when we're designing our experiments. So here's just a general overview of how the retinal responds to the light, right? So we have this hyperpolarization that takes place from the photoreceptor, depolarization within the bipolar cell, and then here's a recording of the action potentials that are going on from the ganglion to the brain. So let's look at the pathway and the understanding of what's going on with the optic nerve. So it's important to think about, so you have this just different pathways, and I just want to use this image to show you that there are many different brain regions, and we're going to break this down a little bit more, that play an active role in what is being processed and how it's being processed. And so, but basically what happens is you have a object that's being seen, focusing that occurs, optic nerve carries in signals into the optic through the optic chiasm, and then information is sent to these different areas, and we'll break that down a little bit more. So here, the primary visual pathway, this is such a beautiful picture. You can see in this, our brain is so, sorry, I get so excited. I love neuroanatomy, but you can see here, this is going to be the right, yellow is the right visual eye, and it crosses over, and the same thing in the green is going to be the left visual side. And you can see the pathways that's going from the eye through the optic, from the optic nerve, and then it crosses over at the optic chiasm, and then going down into the LGN, and then you're going into the visual cortex. So let's break this down a little bit more. And we're going to talk a little bit more and focus on what the LGN does, because it's the main relay visual information from the retina to reach the cortex. And it's important to note that the retina only makes up 20% of all inputs that go to the LGN, and the rest are coming from the brainstem and the cortex. So it's important to think that simply acting as a basic relay for the visual input from the LGN is actually the first part of our visual pathway that can be modified, and it's also modified by different mental states. So let's think about this next part is really thinking about what's the role of the LGN. And the dorsal lateral thalamic nucleus has 6 layers. And we're just going to briefly go over this, but it's important to understand that the different layers have different roles. And so each layer receives input from only one eye. And the inner 2 layers contain these large cells, which are called the magnocellular. And we'll take a closer look at an H&E stain of these layers. And the outer 4 layers contain these small cells, parvocellular. And it's important to think about the neurons for the LGN project through the optic radiations to the region of the occipital cortex, and which is termed the primary visual cortex, the striate. And there's also an area called the extrastriate. So we'll go through that in a minute. So here you can see the visual fields. Here are where like the eye, the fova, it's crossing over the optic chiasm here. And here is a diagram of these different layers, right? So information from the contralateral eye goes to layers 1, 4, and 6. And then information from the ipsilateral, which ipsilateral means the same side, is going to go to 2, 3, and 5. And that's important to think about if you're working with a lot of individuals that have had stroke. You have to think about these things when you're designing rehab programs for them. So here is a beautiful image of these different layers of the LGN, and you can see that these are the parvocellular layers, and then also you have the magnocellular here, and axons from one eye are being labeled in this image here. So the next thing I just want to go through and really think about is the primary visual cortex. And I'm just going to like touch on that a little bit. So you can see here, this is V1, this is primary visual cortex. This is what's called the extrastriate cortex. And we'll talk about that because we have actually a dorsal and a ventral pathway that aids in the processing that takes place. So the primary visual cortex, the left visual field projects onto the right occipital lobe and vice versa. The central part of the visual field is represented posteriorly, and then the peripheral vision is represented more anteriorly within the cortex of the brain. The superior visual field is represented ventrally. And so it's just understanding this mechanism and what is the homunculus of the visual cortex. So the next slide, I'm just going to geek out a little bit because I love actual cells and looking at different layers and pyramidal cells. They're so beautiful. You have 6 layers, right? So here are the 6 different layers. The principal layer of the inputs from the LGN is layer 4, right? And you can see here that layer 4 is actually broken down into 4 subsets. And just noticing the differentials of these neurons and the different, it just tells you a lot about their function. It's kind of like also like teaching anatomy. I'm always like, look at the muscle. What is it shaped? It always teaches us a little bit about what those roles are. And then let's go into what the visual association cortex, which is considered the extrastriate. You can see here that here's the primary cortex. And then in relationship, we have what's called the extrastriate, which is going to have, we'll go into this. There is a actual ventral pathway and then also a dorsal pathway. And you can see here, a lot of us focus on this motor area, the primary motor area here, but it's important to think about the relationship to the Broca area and the primary auditory cortex as well as within respects. So these 2 extrastriate areas, why it's so important to think about is that we have this dorsal stream. So here's the primary visual cortex here in the occipital lobe. And you see here, if you go forward, is the dorsal stream. And that's really important to understand like spatial awareness and visually guided behavior. And here is the second, which is the ventral, which is like the what stream. And this is really responsible for recognition and discrimination of visual shapes and objects and perception and recognition of faces. And if you look at some imaging, fMRI imaging, you can see depending on, I work with a lot of individuals with stroke and imaging, you can see that these different pathways actually have to reassess, and there's a lot of plasticity that occurs over time if one of these areas is damaged and that recognition and perception of shape changes. So just wanted to also further say that these systems are very complex, but I'm really excited to share. This is just first layer of our learning, and maybe we can have more workshops and understanding the integration. And the end goal is to really understand like the multimodal controls, right? Going back to what James is talking about, I'm going to use this paper thinking about grasping a bottle, right? This idea that we have early visual analysis, right? And now we understand what that analysis takes place from a vestibular system and also a visual system. We have visual analysis of the action. We have goals and intention of different parts of the brain. And then we have a reach and grasp control. And then we have muscle control. And I know this diagram makes it a little simple, but I think I wanted to end on this, like in this part of the discussion, to really think about that anywhere in this part of this like pathways and these processing and decision making, if you have somebody that has different, disorders or different abilities within that space, these are all different things that have to be accommodated. And I'm speaking more from a rehab perspective, is that we have to change our goals and intentions based anywhere around this pathway. So I think I have a few more minutes. I will finish up quickly. How do we study these? I just pulled 3 really cool papers that I found, and I'm not biased to Harvard. I think all 3 of them were at Harvard, so I apologize for that, but great school. I'm thinking about just really what are some other models, right? I think a lot of times we forget about how important zebrafish are as a model for postural control because of the simple makeup. So I just wanted to put this out there. I shared my slides too. You can look more. This paper is really interesting. But the zebrafish are a really great model to studying postural control and also organization of their vestibular system because of the simplicity of the organism. The other thing I just want to bring out is, which is really, really amazing that's happening, the technology that's being used in understanding and imaging 3D head orientation movement in the primary visual cortex in live imaging that is happening. Lots of really, really beautiful work that's being done, and I encourage people to look at this work. The other, I just wanted to end, there's lots of different ways investigating visual system and proprioception. A lot of us actually look at grasp or reach, and we do these actually across different models using either robotics or a lot of, I just wanted to bring that forward, that there's lots of different ways to study this information. So I seem to have lost my face. Let's see here. So any questions?
Hi, Kelly, I think that there was a question posted in the chat. It was similar to a question I was going to ask, so I'll read Leia's question.
Okay.
I believe you mentioned that understanding the 6 layers of the primary visual cortex is important to adapt clinical interventions or designing experiments.
Mm-hmm.
So could you give an example of what that looks like? Like how this information could inform an experimental design?
Yeah, absolutely. I'm having some technical difficulties. Can someone stop my share? Like my computer is all
Let me
wonky.
see if I can.
It's actually giving me the
I don't
color wheel. Sorry. So
Oh,
while we try to figure that out, you're probably seeing something really. Are you seeing my screen at all or no?
yes, we still see your screen, but I haven't figured out how to stop your share yet.
Uh,
But if you start, you can answer the question. I'll try to see if I
Yeah,
can
yeah,
troubleshoot.
sounds great. My computer is getting ready to blow up, so I hope I'm still here. Yeah, so I think it's really important because you're thinking about these different layers and the communication that occurs. And for example, if you are not getting, if you know that someone has a stroke that's affecting the upper part of the LGN, thank you so much. You would want to think about, and this is where this is always a big question in rehab medicine, how many physicians actually are looking at the imaging before working with their PMR team to say what regions of the brain are actually damaged, and then thinking about it in the sense of what are we actually going to do. And I think that this is something that we have been really working on with Steve Kramer and this working on remote rehab, is really taking a detailed understanding of what parts of the brain are damaged and how is that going to affect the outcomes that you are going to expect from this patient, and what are things that you need to work on, what are the areas, the aspects of plasticity that one needs to really focus on, if that's possible, if that makes sense. And it's a really, we need to get here. I'm going to give a push. Rehab medicine needs to change. We need to be more detailed and oriented so we can get more precise recovery. But instead we give blanket rehab strategies across. If you've had an MCA stroke, okay, we do X, Y, and Z. And I'll say to you that looking at imaging and understanding what parts of the brain are actual injured, then you can really do a more detailed assessment, if that makes sense.
Yeah,
I'm
related.
very passionate about this. We really need to
Yeah.
change the way we do it. And in England, this is what happens. In other countries, in Sweden, they look at imaging. Why are we not doing it in our country? I don't know. It's
So that would
expensive.
be more individualized care then,
Yeah.
basically, for each
Mm-hmm.
individual. Yeah. Sorry,
Yeah.
Jill, you were saying something? Jill, were you going to say something?
Yeah, I wanted to also emphasize what Kelly brought up about really learning from the patients, and in particular with like Paralympic athletes and adapted recreation, because they are involved in very clear goal-directed tasks, and they are also practicing and learning. And so this idea that also brought up, I think James also brought this up, is it volitional or are you just reflexive?
Right.
And so really thinking about personalizing the intervention is working with this particular population. They're really motivated to learn and adapt. And if you spend some time talking with someone about, again, what they have and what they use and what they've been able to muster through the really specific training, it's amazing what the outcomes are. And I think studying that population can really help rethink what we're doing in rehab to get this personalized care. So it just happens to be a very unique population, but partnering with them and learning what they've learned has helped, can also inform that care for others. So I just wanted to bring that up.
Yeah, absolutely, Jill. Thanks so much. I just want to to add like one more thing. I think it's really important when you are, so for example, I'm running a Wounded Warriors project right now in Mammoth, and I'm working with different, like all different disorders, but we're doing adaptive sports. Every single person I'm working with is different, and we're having to reassess. And also, what's really important to think about, going back to what James was saying, is they have all over time have different adaptations, right? And really reassessing and adjusting what are those adaptations that are taking place. And I would love to do, imaging and all of these individuals and see like what are these adaptations. But yeah, it's really important that we look at the whole picture.
Any questions here? Other questions?
I'll just add a couple of points here that I wasn't able to sort of include. So, we had sort of a 2 hour and a half together today, and this is typically several lectures or discussions in, let's say, a neuroscience course. So we compressed a lot here. So one thing that's important that I don't think we really touched on is really when we think about sensory motor control, a lot of these sensory systems, they actually converge. And so what you end up with is what we call multi-sensory processing. So balance control is, I think, the perfect example where we've all experienced this, right? If I stand on one leg, I can maybe do that. If I close my eyes, some people are better than others. And that tells you a little bit about how much you use vision for balance, right? Some people are really good at that because they're relying primarily on proprioception. And so, vision, vestibular information, somatosensory information are all involved in things like balance control. And if one of those systems goes offline for whatever reason, whether it's disease or some experimental manipulation, you can actually learn to reweight those systems and rely more on one versus the other. So, for instance, if I have like what's called chronic ankle instability because I sprain my ankle every year I play basketball, so I have very poor proprioception in my ankles. So if I close my eyes and try to balance on one foot, like I fall almost immediately. But if I were to train, I could sort of relearn how to use that information that's coming from the periphery. So that's about multi-sensory integration, which I think is really fascinating. The other thing that maybe relates more to IMSI world here, there are a lot of really cool studies. So Richard Nichols is an example of somebody whose work that I think might interest a lot of people here. And what he's done a lot of is look at what happens when, for instance, proprioceptive information. So you can actually, in an animal model, certain animal models, you can transect the nerve that comes, the afferent nerve that comes back to the spinal cord. And what this does is it can prevent the animal from using, let's say, muscle spindle information. And then you can look at what happens to the behavior over time in the absence of that information. So that's this idea of using a lesion model is a really fundamental tool in neuroscience and behavior to try to understand how organisms use certain sources of information. So that's another form of a perturbation. So I think a lot of the work that we're doing in IMSI uses perturbations of various types. But if you think about, there's a perturbation, an immediate effect of that perturbation, but then there's learning. And so I think that's something to always keep in mind in all of our studies.
One thing that you brought up is that, you know, when we're talking about rehab, there's a huge amount of individual variation, but even in healthy populations, the system's so complex, right? Like James, you have ankle instability. I also have had many joint injuries over the years. And so I have all kinds of instabilities. If I close my eyes, it's a mess, right? So I clearly have to rely more on visual information, right? So when we're designing perturbation studies, might have this really careful experimental design, but then our individual subjects bring complete like systems that are so complex and different, right? So I wonder if you have thoughts about how you deal with individual variation, because often in these studies would like look at the group mean average, but that's hiding a lot of really interesting information about people's individual responses, right?
Yeah, I think about this quite often. And actually, this came up in one of my former PhD students' dissertations, and Jill was on that committee, and we've had some of these conversations. So, since Tara's on here, I'll use Tara's work as an example. So she's been looking at how people deal with predictable versus predictable perturbations during walking, and you give people lots and lots of perturbations over the course of an experiment. And so one thing that we often like to do is you can start with looking at your individual participant and how behavior changes over time. And so that tells you something about, whether they sort of learn or whether they are highly variable. So I think that's important. Another thing that we've done in another set of studies from a student who recently defended, you can think about many of the kinds of experiments that we do as like we're trying to kind of phenotype a person. We're trying to see how they behave. But one critical aspect of that is whether that behavior is consistent or reliable. So you can do things like test if there's not a learning that's involved in the task, you can test them on one day, bring them back a week later, and then see if your outcome measure is consistent. So I think that's something that is underappreciated in most of the work that we do. But now, if you can show that like these two people are different and they're different on multiple days, now you can be a lot more confident that you're not just dealing with the random variation that you see in a person from moment to moment.
But then if we have studies where now we have maybe 20 subjects and 20 slightly different kind of response patterns to a perturbation, how do we then try to draw conclusions and make useful inferences about control mechanisms from that?
Yeah, so it depends a bit on the exact outcome measures. So in our work, there's good and bad about this, right? We often will look at group effects. So we'll say this group of people who've had a stroke or older adults or this group of young people, and again, this is comparing means, but you lose enough information when doing that. What I think is most important is to try to think carefully about sources of variation that might introduce variability in whatever your measures are, and you control or measure it, right? And so for us, we always struggle with do I use older adults versus young adults, or do I treat age as a continuous variable? Well, you can dig another layer. It's not actually aging. Yes, we age, but it's not so much that people are older, it's that it has changed as age. So what is that something? Let's measure that and see if that better explains behavior. But I think that's an important aspect of studying whole organisms, definitely. But so things like people's height, if you're doing treadmill perturbation, how are you normalizing speed of the perturbation to their walk? Those things can help reduce variation if you can identify where the source of the variation.
Yeah. The one thing we've talked about in the context of aging is like, well, is it aging or is it activity level, right?
Yes.
With aging comes inactivity, often they're very coupled, right?
100%. Yeah, the same student did the test retest study, we did this study, we found no difference among, older adults who were largely 65 plus and a set of younger adults who were largely graduate students. But our older adults actually happen to be more active than the younger adults because
Right.
of our, we're recruiting from this database. It's LA, right? So we have this sort of sampling bias, we think. And so I do think things like activity level are more important, can be more important than age, whatever that tells us, right? So
Right.
yeah, there's a whole world of studies I think that have missed that issue.
Yeah.
Yeah, and also thinking about previous experience,
Yeah.
right? Like the previous experience is, we see that a
Training
lot
and
with
experience.
dancers.
Yeah.
Yeah, I
Yeah.
mean, everyone thinks every dancer's trained the same way. That's absolutely not the case, you know?
Ruby has a question here
Yeah,
too.
so going back to comment about imaging, how can that help potentially give a bigger picture on how variability presents itself amongst individuals? So I just want to kind of get like an example of how, if you use imaging personally to give you a better indication on how these variations came about. Does that make sense?
Can you hear her question okay?
Yeah, I think that question is for me. Yeah, yeah. I just want to make sure. So yeah, absolutely. I think, and I did a study with Dr. Kramer when I was doing my postdoc, and what we literally looked at was that we did stroke volumes on and found out where each stroke was and what pathways were affected during the injury, right? And then actually did a stroke volume measurement, like how big was the stroke and where was it and what pathways. And then we did tailored rehab. And I'm not saying that we can do all, it's very labor intensive. But what we then did was looked at the different pathways that were injured with that stroke. And then we tailored the rehab program based on what if it was a like more of a premotor, right? Like thinking about more before the decisions actually being made, we implemented a different strategy versus if it was just the motor area, right? And then a lot of times, so if it's depending on the side of the brain, we actually had to change our whole process if, for example, part of the visual cortex was taken out as well. It's just we based our rehab plan on what injuries were, what part of the brain was injured, and then also looking at what was the functional output of these injuries because not everyone's the same, right? It's really if for example, if a stroke has taken out the CST or a cerebrospinal tract, like the recovery is also going to be different. So really kind of looking at what parts of the brain were injured and then going and doing rehab. Now, do it's going to happen like that we do this all the time? Absolutely not right away. It's extremely laborious, but it really from an experimental design perspective, also it's important to understand it gives you a basis to know what is missing or what and then also the plasticity that takes place, right? We notice that in our acute stroke individuals, there's a very interesting time period between week 7 and 12 of recovery, and there's also different bivariants that aid in that recovery time period of where we need to do the intervention, if that makes sense.
Yeah, no, it's just, I was mostly curious because I think it's great because it definitely gives you more information that's very
Yeah.
necessary for interventions. It was just
Yeah.
thinking like more broader picture. Like I just started thinking about how complicated it is with insurance. And that's like the frustrating bit within PT. So yeah, I was
Yeah.
just more curious. But yeah, experimental
No,
design, 100%.
I didn't say that it was functional in our current medical system.
Yeah.
I mean, this is why rehab medicine is so hard to get money for too. Like we would love to be doing these things. And we do have 2 trials going on at UCI that's doing more of this tailored rehab strategies at our hospital, but I would love to talk to you more about it. But it's definitely it's really fascinating.
Yeah.
I think you mentioned that people are doing this more in other medical systems, like in Europe. So is that because they have a more socialized medical system, or what's the difference there that allows it
I
to be?
mean, yeah, it's the more socialized aspect, right? And it's really, you know, that's a
Yeah.
larger conversation. But yeah, absolutely. So, and then I'm trying to get work that I'm trying to work with somebody actually in Sweden right now, using some of the things I did in my postdoc, like doing some more individualized and a lot of individuals too, like I mean, a lot of tele-rehab we started tele-rehab way before COVID, but, tele-rehab, is really, really becoming tailored and really important because it just really adds a lot of access, right? Most of us live in large populated areas, but a lot of individuals do not, right? And giving access, like tailored healthcare is really important.
All right. I think we're kind of
I know we're over.
over time a little bit.